Okay, so the next cell type we're going to look at are the monocytes and macrophages. So neutrophils and eosinophils are more of the kind of generalized phagocytes. Monocytes and macrophages are more of a specialized type of phagocyte. So you can think of them almost as like an elite type of white blood cell. Now, these two cells are connected in the sense that they actually begin as monocytes, and then monocytes will transform into macrophages. And so this takes place through various stages of the development, cell development. Cells start out as a stem cell. They ultimately will develop into a monocyte. Monocytes then are somewhat characterized by being found in the bloodstream. And monocytes then will become macrophages after they migrate out of the bloodstream and into the tissue. So you have stem cells in the bone marrow, which in this particular sequence will develop into monocytes, which travel through the blood, and then ultimately migrate into the tissue for their final stage of development. Now, really what's happening here is the body is creating monocytes in preparation for the need of macrophages. So what will happen is monocytes will circulate in the blood until they are stimulated. It says right here, due to chemical stimuli. So occurs, monocytes transform into macrophages after they migrate out of the bloodstream into the tissue due to chemical stimuli. And this has to do with other cells that are signaling them to come in to that area, which we'll talk about that later. So during a chemical stimulus, they migrate out and become macrophages. These are kind of like elite cells. You can almost think of a monocyte as like a cell in training to become an elite cell. And the macrophage is that cell at its kind of end of that elite training. So macrophages then are somewhat differentiated by the fact that they're no longer in the bloodstream, they're found in the tissue. They're larger in size, and they have an enhanced development of lysosomes, primarily two other organelles like peroxisomes, but lysosomes in particular. And if you recall from chapter 5, we talked about lysosomes being an organelle for digestion. So for example, I mentioned that these are kind of like the stomach of the cell, and they are specialized in digesting things. So in these cells, that capacity is somewhat tailored towards phagocytosis and breaking down harmful pathogens. Now, there are a variety of different types of macrophages. So there's some different terminology here that will reference different particular tissue types. So we have the monocytes, they become the macrophages. In many cases, we simply refer to them as macrophages. When they're found outside of these tissues down here and not in the bloodstream, we call them macrophages. Okay, so these examples would include the spleen, lymph nodes, bone marrow, kidney, bone, and brain. And so sometimes these macrophages travel back into the bone marrow after development. As I said here, they begin in the bone marrow. Sometimes they can travel backwards. So we refer to them simply as macrophages. Now, histiocytes are a specialized type of macrophage, and these are often characterized by where they're found, in addition to the fact that they often have some extra unique features. But examples include the alveolar macrophages. Alveolar macrophages are found in the lungs, alongside the alveoli, part of the lung tissue. So these are called alveolar macrophages. Kapuffer cells are found in the liver, named after Dr. Kapuffer, who named them after himself. And they're found in the liver. Dendritic cells are found in the skin around the dermal layer. So these are cells that have evolved to take on a special role in areas of the body that tend to see high amounts of pathogen or antigen come their way. So if you remember from chapter 13, we said the lungs are the most common portal of entry. So you have lots of microbes coming in there. So these cells are specialized to kind of play a role in that kind of high volume area, so to speak. The liver is a kind of a filtering organ. Lots of things come through there, and then the skin is susceptible to damage and all sorts of infection. So have specialized cells there as well. So those are just some different terms that apply to different locations in the macrophages there. Now, next thing I want to talk about here is a special role that macrophages play. Macrophages are a key cell that bridge the gap between the second and the third line of the immune system. So earlier I talked about the 3 levels of defense, the second level being the non-specific and the third level being the specific. Here's that diagram here. So we've got the second line and the third line. What macrophages are going to do is they're really going to trigger a connective kind of bridge, so to speak, between these 2 levels here. So these don't operate in a vacuum. They all tend to kind of overlap at times, even though we're kind of describing them as 3 different levels, or maybe think of them as layers. They're not totally separate from each other. So they definitely overlap and interact. That's part of what you read in the chapter 14 textbook questions. They're part of the textbook reading. So there's different ways that they can interact, and so we're not going to talk about all of those, but one of the common ways that the second line ultimately leads to a third line response is through the activity of macrophages. And really, it typically involves a cell called an antigen presenting cell, APC. But most commonly, these are macrophages. So there are other exceptions to the macrophage being an APC, but typically it's a macrophage. So we're kind of focusing on the most common scenario here. So what's going to happen is ultimately you're going to have some kind of microbe pass the barrier to infection. They're going to go from they're not going to be stopped here in the first layer. They're ultimately going to get into the second layer here. And that's going to trigger a response to a non-self entity. So the body is going to recognize there's a non-self entity there, and it's going to attack it. Now, what often will happen first is neutrophils, and for neutrophils, they will often be the first responder there. Neutrophils, going back here just real quick, neutrophils will send out chemical stimuli that trigger monocytes to come into the tissue and become macrophages. So neutrophils ultimately trigger the macrophages. Macrophages then act as an antigen presenting cell, and what they do is they start to gobble up some of the microbe. And when it comes to their roles in antigen presenting cell, they phagocytize, so they gobble up this foreign microbe, and then what they do is they break it down inside the cell, and they literally take parts of that microbe, little pieces of it, and they display it on the outer part of their cell. Then they take those many little pieces of the microbe, and they literally travel to the third line cells. In this case, they typically activate the T cells themselves, and the T cell activation then triggers that third line of defense to begin. So we go from a bypass of the barriers. Typically neutrophils are activated, triggers macrophages. They act as an antigen presenting cell. As an antigen presenting cell, they carry the antigen and they activate T cells. Now what happens next we'll talk about in chapter 15. T cells will activate B cells will create antibodies, and they all kind of act in concert at that point. But you can kind of see the connections here is what's happening. So barriers fail, non-self is recognized, typically a neutrophil leads to the activation of a macrophage. And by the way, there's more things here. I'm summarizing. I'm not talking every detail. There's not always neutrophils that have to activate the macrophages, but that's a common way. Macrophages phagocytize, acting as an antigen presenting cell, carry the antigen to the T cells and the B cells, and that activates your third line of the immune system. Now, just one more note about this here. Let's just stay here actually. These cell types here, the B and T lymphocytes, they tend to be found in two key areas of the body. Now, this is prior to their activation. So before they're activated, they tend to be found in the lymph nodes as well as the spleen. So these cells, before activation, are not just out patrolling the body like we see cells of the second line of the immune system. So these cells are really patrolling, scanning the tissue. If you remember just go back here real quick to remind you, some of the roles of phagocytes are surveillance. So they're out patrolling the body. It's not really true of the B and T cells. They're only activated, they're only in the body tissues after they've been activated. So prior to that, they're not actually scanning the body for the most part. So they have to actually be kind of woken up, so to speak. And so they're kind of sequestered away, and the way that they get activated or woken up is by the antigen presenting cell bringing the antigen, parts of the microbe, to the T cell. And then that triggers a variety of responses thereafter. So that's a key part of how the overall system works together. And the triggering of macrophages is the most common way that the third line is activated. And sometimes, to be fair, sometimes antigens can kind of bypass the first barrier and immediately trigger that third line. So, for example, something will just drain into the lymph nodes. So sometimes antigen from a microbe will simply trickle into the lymph nodes and activate these cells independent of a macrophage. So there's a variety of ways the immune system works, but most commonly it goes through these layers here, from barriers to neutrophils to macrophages to T cells to the third line being activated. Okay, so next I want to talk about some of the more specific events in phagocytosis. So we've talked about what that is, and now we're just going to talk about it in a little more detail here. So first of all, microbes are activated through what's called chemotaxis. And chemotaxis is defined as movement in response to chemical stimulus or stimuli, something to that effect. And this really applies in a variety of different contexts. Sorry, I'll leave that up there for one more second. Chemotaxis, movement in response to a chemical stimulus or stimuli. This is something that can take place in a variety of different contexts. So it's not specific to phagocytes, but in this context, it can apply to phagocytes. So there's other ways that you can use that word in different contexts. But in this context, what it's referencing is the fact that your phagocytes are literally chemically attracted and move in response to stimuli given off by microbes. And you know what, I've got a really cool video I'll show you that will illustrate this better than I could ever explain. Okay, so this is off YouTube here. The title is called White Blood Cell Chases Bacteria. Fairly popular video as it looks here. And to point this out here, you have red blood cells here. These are red blood cells. This right here is a white blood cell. It doesn't say what kind it is, so I'm not exactly sure the exact type, but it is a white blood cell. And then right here, these two little black specks, these are a type of bacteria, appear to be a diplococci. Okay, so watch what happens next. So you can see that the white blood cell is literally chasing the bacteria. And this is taking place through chemotaxis. The white blood cell is almost like a dog sniffing out some kind of rabbit and chasing after it. Now it's you don't see all the details there, but what had happened there is it finally grabbed a hold of it. Grabs a hold of it right about here and then quickly phagocytizes it. This is a very low resolution microscope, obviously, so we're not seeing all the details there, but that is a kind of the sped up process of phagocytosis. Now this, by the way, is a stop motion image, so it's being fast forwarded at a high speed, so it's not something that would take place this fast in reality, but in a kind of slow motion you would see the same thing playing out in reality. So chemotaxis looks just like that. It's your body kind of chasing after, so to speak, these microbes. Wouldn't always have to do that, by the way. Sometimes it would grab them much faster. So chemical response there. Now the next step, I actually left it off the bullet point here, so let me point that out. The next step is actually adhesion of the bacteria. So it goes from a chemotactic response. Once they grab a hold of them, they adhere, and that involves the adhesion. So the bacteria have to stick to the cell. If you remember back in chapter 4, I said that this is one of the problems with encapsulated bacteria, or bacteria with the capsule. It makes that process more difficult. They can't grab a hold of them. So normally speaking, they would grab a hold. You have these receptors here that grab a hold of them, and next thing is the cell swallows them, and that's the ingestion stage. So chemotaxis followed by adhesion, then engulfment, and the engulfment involves the membrane kind of sucking them into the cell. This then encloses them in what's called a phagosome. And a phagosome is basically a specialized vesicle that entraps these microbes. So this membrane invaginates backwards, seals itself off, and now creates a specialized type of vesicle called the phagosome. Now inside the phagosome, the bacteria are technically still alive. Nothing's really happened to them. They've only just been kind of sealed up. The next step is critical to their death, and that is that we have these lysosomes that we mentioned, key parts of macrophages, the digestive organelle of the cell. And the lysosome is literally going to fuse with the phagosome, and that involves then the formation of what's called the phagolysosome. So these two things fuse together, and this is kind of like dumping a bunch of toxic chemicals into the phagosome. So inside there with all these chemicals in, most of the microbes, if not all of the microbes, are going to be immediately killed, and this is a very effective effective concoction of toxic chemicals, and it will kill any microbes in there. So they're broken down, they're killed, they're degraded, chopped up in little pieces, so to speak, and in many cases they're excreted, which is the excretion of these bits and pieces now outside of the cell. So it's almost like a person, so to speak, or any animal eating, swallowing, digesting, and excreting. It's happening on an individual cell level. Now, one difference here would be if we're looking at an antigen presenting cell, one difference would be that rather than excreting everything, some of these components would actually be routed to the Golgi apparatus. If you remember from chapter 5, the Golgi apparatus is like the UPS of the cell. It's involved in packaging and shipping, and some of these pieces here would actually be packaged and shipped back to the membrane where they would be displayed for antigen presentation. So that part's not shown here, but that would be another kind of side event that would take place with macrophages. Okay, I'm going to post a link to this video online, or download, that is. You don't have to watch it, but it's just a nice little visual there of what's happening there. So back to the concept of chemotaxis. Chemotaxis is something that often takes place in response to what are called PAMPs, pathogen-associated molecular proteins. So if you think of the white blood cell as kind of like a dog, and the microbe as kind of like some kind of rabbit or squirrel, or maybe a raccoon if you're talking about a coon dog, then the PAMP is the particular smell or the particular characteristic that the dog will be picking up on, smell. And that's really kind of a way to think about it here. So, and this really kind of gets into an evolutionary context, because really what's happened over years of evolution is that our immune system has evolved to recognize things that are deemed to be foreign and harmful. And really what's happening is the immune system is profiling against particular types of recognized pathogens. So our immune system has evolved to key in on pathogen-associated molecular patterns, PAMPs. And so just to go back here and connect it to what I said earlier, this idea of non-self here, PAMPs are very distinct, recognized aspects of the non-self. So this is a complex thing. Non-self can take on many different contexts, and there's lots of different things you can get into here. But one of the most widely recognized concepts of quote-unquote non-self are these PAMPs. In other words, the body automatically recognizes them as being non-self. And that is, again, kind of based on a profile mechanism. So ultimately what they do is, there's something that the pathogen, that the phagocyte recognizes as being foreign. They serve as a signal molecule. And the key thing here is that these are not present in mammals, where our immune system takes place. Obviously, we're a mammal, we're talking about the immune system here of a mammal. And so the key thing here is that these PAMPs have to be something that wouldn't be mistaken within our own body. So examples of PAMPs that you don't find in animals that would be exclusive to microbes would be peptidoglycan. Now think real quick, do you remember where peptidoglycan comes from? What about lipopolysaccharide? If you can't remember, these two things here both come from bacteria. Peptidoglycan is the primary component of a bacterial cell wall. That is where peptidoglycan comes from. So this is a cell wall component only found in bacteria. It's a molecule exclusive to bacteria. So by recognizing this molecule, the immune system can automatically identify anything with this chemical as being a bacteria, ultimately being of a bacterial nature. The other one, lipopolysaccharide, do it backwards this time, lipopolysaccharide. This exclusively comes from a gram-negative, this way, outer membrane. So the outer membrane of gram-negatives, we said the outer membrane is a key part of gram-negative bacteria. And this outer membrane is made of a certain chemical. This is just part of the outer membrane, it's not the entire thing, but there's a key chemical in there called lipopolysaccharide. And that chemical is only found in gram-negatives. So when the immune system recognizes either of these two chemicals, it automatically can associate them as being foreign, not being part of our body, therefore being non-self, and it's an immediate trigger that kicks off that phagocytic process. Another example is double-stranded RNA. We didn't talk too much about the different types of viruses back in chapter 6, but I did mention that there are RNA viruses, and I'll go ahead and clarify here. RNA is always single-stranded, with the exception of double-stranded RNA viruses. So these are literally the only time in nature that you will find RNA in a double-stranded form. Now DNA is double-stranded, but RNA is always single-stranded. Now we haven't talked about that yet. That's a part of chapter 9, so I don't expect you to know that, or I'm not really going to ask you about that for any quiz or anything. But other than maybe I might ask you as an example of a PAMP, but I don't expect you to know the innate details of RNA at this point. But just to clarify, RNA is always in a single-stranded form, and DNA is always in a double-stranded form. Real quick, just to give you a visual, here's the side-by-side comparison. RNA is a single strand, DNA are two strands joined together. So the only time you would find a double strand of RNA, as I mentioned, is with a type of virus. So if the body sees that and it knows that's a virus. These are a few other examples, just to go show you a couple of visuals here and show you kind of the range of these. I pulled this off the web here. You have different types of PAMPs found in a wide variety of different microbes. So viruses can give off several different types of PAMPs. Gram-positive bacteria give off several different types of PAMPs. So all these here, like GP, DNA, RNA, DNA here, all of these are examples of PAMPs. Now they're all in acronym form, and this is a different figure for a different textbook. So I'm not going to use this for on the test or anything, but just to show you some of the different examples here. These can all be recognized as a PAMP here. Now the way the body actually recognizes the PAMP are through what are called pattern recognition receptors. So this kind of goes back to the lock and key mechanism that we find in many examples in biology. So I talked about it in enzymes and substrates, I talked about it with viruses and host cells, and once again we see it here. So the PAMP is like the key, and the pattern recognition receptor is like the lock. And in this case, the PAMP has to bind to a pattern recognition receptors. Now these are a little more general, not quite as specific as some of the lock and key mechanisms with between viruses and host cells. They're more like a general key. It's almost like if you work at a building and they give you a key to the entire 5th floor, for example. That's kind of how these work. They're not quite as specific as other types of lock and key interactions, but they're still that same general kind of mechanism here. So these pattern recognition receptors are often found on phagocytic cells, dendritic cells, especially a type of macrophage, and then even some of our endothelial cells will have these as well. And that gets into a more in-depth part of the immune system that we really don't cover here. So, but these are basically the way to actually bind to the PAMP. And if we just go back here real quick, you see here that that's what's happening in immediate response to chemotaxis. So the chemotactic response allows the microbe or the phagocyte to adhere to the bacteria, and they're doing that with the pattern recognition receptor on the host cell. So that's actually how they lock onto them there. An example is of a specific type is called a toll-like receptor. These are the most common type of pattern recognition receptor, and so they are what specifically grab a hold of these microbes here. And if you just go back and look at this, TLRs here was showing how these different toll-like receptors bind to these various different PAMPs here. Okay, next we're going to talk about the inflammatory response.